Hello, everyone. Welcome to AgBioData webinar. I'm very happy to introduce Seth Murray, today's webinar speaker. Seth is a professor and Eugene Butler Endowed Chair in the Department of Soil and Crop Sciences at Texas A&M University. He leads a maize breeding program and develops approaches in drone-based field phenomics, quantitative genetic discovery, and data analytics. He currently serves as the past president of the Crop Science Society of America. I welcome Seth to present his work. Seth, here you go.
Okay, well, thank you. It's great to see some past friends and some people I don't see very often. So this is a very different audience than I normally present to. I tried to change my slides around a little bit, knowing a little bit about ag biodata, so focusing more on the data we're collecting and a little less on the science and the biology. But if anybody wants to discuss that, I'm happy to. And I'll also apologize. I was in bed for 15 hours yesterday with what I presume is COVID, and I'm still recovering. So I'll try and be as enthusiastic as possible, but I'm struggling a little bit here. And this slide here just sort of represents we typically are flying drones now over all of our fields, and I think over most fields this year we got about 77 flights was the number I was given yesterday across everything. So drones have become an integral part of the science we're doing, and it takes a big team, and that's why the et al is so big, and I'll cover that at the end. I'm just going to give you a couple background slides here, just based on what I do and sort of why my position exists. I'm focused as a corn breeder. Every land-grant university in the United States used to have a public sector corn breeder. Of course, industry has taken over much of that. But I'm based here in my pointer, based here in Central College Station, Central Texas and College Station. But of course, most of the maize that's produced is grain maize for feed and fuel, and that's produced in the Midwest. So we have a very different environment here, which creates some different challenges. And just to give you an example here, the yield of maize has continued to increase, and then the red line here is Texas as the irrigated high plains came online. And then the area I breed for is this, which tends to be the non-irrigated acres. Our yield is less than half of the U.S. average. So a lot of different stresses, especially aflatoxin or Aspergillus flavus that's produced. But we do run an applied breeding program. So we've developed a number of lines. And you can see here just in an open pollinated commercial expired plant variety protection line, we have poor seed set, just so much stress. And then probably the thing our breeding program has become most known for is developing varieties for whiskey. So another little piece of background, because I recognize this group is mainly probably not plant breeding based. We've had a big shift in plant breeding from sort of this breeder's eye, which is very subjective and expertise, to this objective measurement of things we care about. And I'm guessing most people here are familiar with QTL mapping and GWAS and genomic prediction and selection, but there's been a lot of other innovations that have occurred. And I think the next wave of innovations is on the phenomic side, and that's what I'll primarily be talking about today. But this breeder's eye, or this subjective evaluation that people made to make all these early gains in plant breeding in maize, I think that's really what we're doing, quantifying things. And I'll try and touch on that at the end about what this phenomic approach is doing. I think it's always important to mention that yield is what we mainly focus on in maize, but there's a number of other traits now that are increasingly important, and especially talking about climate change, talking about human health, talking about farmer profitability rather than just yield. So all these are things that we're trying to select on now. And the reason that is important is because these are somewhat of phenotypes, right? So a phenotype is what everybody is probably familiar with, talking about plant height, talking about yield, talking about maybe you can measure nitrogen use efficiency, right? Those are single traits. And that's the way most people envision phenomics and high throughput phenotyping. But I'm really hoping to shift your mindset on this much more to the open architecture that we have in something like genotyping by sequencing, where instead of focusing on a single gene, which would be equivalent to a phenotype, we're focusing on the phenome, which is equivalent to this open architecture of saturating the entire genome. So there's a little bit of a linkage there. I found that language is a really important thing here. Phenomic selection, which I'll talk about, is not the same as classical phenotypic selection, and I'll hope to explain that a little bit as we go through. We also find that this language we use when it comes to drones, all four of these terms, five of these terms are in the literature, and we've already documented at least two examples where somebody has said, nobody's done this before, and when we follow up with them, it's because they searched for one of these terms and not all five of them. So this language is really becoming problematic in this area. And that's really nothing new. Multidimensional scaling and principal component analysis, those developed in different areas, and they're essentially the same approach. So as we have all these different disciplines developing these tools, we're going to see a lot of issues with language. And I put this slide back in. I leave it out in most presentations because I think this group is one that's really well targeted to sort of address this. So contrasting the phenotype is the phenome. So the phenome is currently an ideal, right? The totality of every possible measurable and estimatable physical characteristic. Okay, so that includes things that we have never conceived of before. My favorite example is plant texture. It's something you can see in an image. You can see that two different plants have a different texture from drone imagery, but we don't know how to measure that. So that's not really a phenotype, right? It's a component of the phenome. So I like to draw a lot of parallels to genomics because, especially knowing this group, I think genomics has been a large focus. And if we think about the history of genomics, phenomics is sort of a little bit more advanced than 1986 in genomics. Not much, though. So if we think about QTL linkage mapping, the goal there was initially that we would find the genes in the genome that condition phenotypes we care about, and we would use those in crop improvement. And it generated a lot of knowledge and a lot of publications, but I struggle to point to too many examples where it's actually resulted in meaningful crop improvement. GWAS, similar goal, different type of statistics and different type of population. Same thing though, a lot of knowledge and publications, not very much crop improvement. The big philosophical difference, and this is the shift to phenomic selection from high throughput phenotyping, is genomic selection. And in there, we're focused on finding the best individual. That's resulted in a lot of crop improvement knowledge, but it's pretty hard to publish on genomic selection because the inferences are specific to that population, that environment, and as industry is developing these, it makes it difficult to add in new germplasm or new types of environments because these predictive equations are so based on past data. So I want to spend a couple of minutes here. High throughput phenotyping. Again, I'm going to belabor this point because I don't think this is still a common way that people are thinking about this. When we first started, it was all about automating routine measurements. So plant height is a great example. The things in green, I'll definitely show you a few examples of. In sorghum or wheat, where the panicles are out, you can actually directly estimate grain yield, or cotton. Estimating disease, there's been a number of examples of that. And so that's scaling routine measurements we're already taking. I think the more exciting thing when you have a new tool is to find new things. So finding new signatures of eliteness, so temporal growth patterns or biomass production, senescence and grain filling periods. And these really resulted in us focusing on this phenomic selection aspect, which hopefully you'll get the gist of as we go through. Now, all three of these rely on tabular data, which any of you familiar in genomics is probably pretty familiar with. But there's this other area developing of deep learning and AI tools directly using images. And those are usually to develop a single phenotype. They're not necessarily connected to the phenome yet. But that's a really exciting area to be a part of. Now, anything we identify through these four areas are also probably relevant for identifying stress signatures for farmers management. And the thing we found a lot is new mechanisms of biological importance, just stuff people weren't previously looking at. To give you an example of scale, though, it's a little bit different when you start, when I first started the drone projects, I was told, oh, these things are easy to do. And once you start talking about we have 17,000 yield plots, all of a sudden, those methods that you can manually do become much more difficult. So we've had to automate a lot of stuff as we've gone through. One other piece of background that I think is important here is we did some simulation studies based on what we were seeing, and both simulations and empirical data has demonstrated that high throughput can actually produce better decisions than high accuracy when we're phenotyping. So as we move to these tools that can actually scale the number of individuals, even if there's more error associated, and I'll show you oftentimes there's not, there's less error associated than in manual measurements, we still can make better decisions because we're scaling it to more individuals and larger populations, more environments. And that's a really big goal as we're talking about crop improvement. So this is a fairly old slide. This is when we first started our drone work in 2016. It was a huge transdisciplinary project. People from agricultural engineering, aerospace engineering, ecosystem sciences, statistics, trying to figure out how we can start to use this data. And again, doing one phenotype in one small study is not challenging. It's when you start to scale this, the way we think about scaling genotyping, like by genotyping by sequencing, and to thousands of individuals. So in brief, we place some ground control points in field. We fly the UAS. Now we have undergraduates doing that. That's actually one of the easiest parts, UAS or drones. Downloading the images, do some spectral correction. And then this ortho-rectifying and mosaicing images, I think is an important step for this group to understand. That's like stitching on your camera phone. There's tie points that are identified, and it creates a mosaic of all these hundreds or thousands of images on a field, because in any one image, we can't identify what's in that image without orthomosaicing it. Although there's a couple alternative methods that can back position, we need to stitch these together to generate the entire field. This algorithm is imperfect. There's a couple different suppliers of various algorithms, and they result in different types of analyses. And so as I think about ag bio data and how those of you who deal with making data fair and open and, how we're going to deal with this step will change over time. These algorithms will continue to improve, and the software you use to do it will make a big impact on the end product. So the images are definitely critical data to maintain, but then for repeatability of an experiment, for confirming somebody's results, you have to know exactly which orthomosaic algorithm they used. We use structure for motion to make 3D point clouds, so that allows us to identify structural features from the plant, like plant height is the primary one. From there, we will identify plots, identify the ground, extract various vegetation indices, and then only at this point do I, with my skill set, know how to do any of this, because everything else is colleagues, collaborators, and my graduate students and postdocs. So this is the data that I'm ultimately interested in here at the end for making better breeding decisions or discovering biology. But all this stuff takes a lot of work and shouldn't be underestimated. So just another old example here. So when you stitch together an orthomosaic, you can see here this orthomosaic on the right has some issues, right? And the method by which you fly really ends up resulting in different types of data. So we often see heritability from one flight to the next flight may remain the same, but if you have a flight in the middle, the heritability may increase or decrease just based on the amount of wind or some issues with lighting or whatever in the flight. So all these things play a role in the ultimate downstream analysis and shouldn't be taken for granted. So this is our current tool, our current pipeline. You can see we use a number of different products. These are not well integrated. Metashape, our UAS tools I'll mention in a second. Field Image R has been absolutely game-changing for us. Felipe Matias and others developed that, and I'll show you an example of that. But there's a lot of different disparate software packages that are used to create these end products that we then use or share with our colleagues and collaborators. This RUAS tools is something I wrote code for just to use internally, and then Stephen Anderson, a grad student with me, turned it into an R package. And what this does is, based on two GPS points on the side of the field, a breeder can connect their field book and develop shape files. So these shape files are important to extract the features within the orthomosaic. So this sets those GPS boundaries that can then be used for extracting the things we care about. So I'll show a couple examples, try and whet your appetite for how this data is being used. Really old study here. The important thing is to show first of all, two different UAVs produce different results for plant height. The other is, this is a dryland field, an optimal planted field, and a late planted field. So you can see that you have different results based on your field environment, as you would expect. And then there's a number of genotypes here. Unfortunately, it looks like, are the slides cut off for you guys at the bottom? You should be able to see something under correlations to grain yield. I don't know why they're.
Yes, it's cut off. Yeah.
Okay. Well, I apologize. In my environment, grain yield is highly correlated to terminal plant height, which we measured with a ruler at the end. But what we found is that by using these drone flights throughout growth, we can actually get better correlations to grain yield. And then these various colors just show different genotypes, and you can see the genotypes behave differently. And importantly, there's some crossover effects. So the genotype that's tallest at the end is not necessarily the genotype that's tallest earlier in growth. So that's when the light bulb went off and said, hey, this temporal aspect, this aspect of measuring plants throughout their growth is really important. So Stephen Anderson took this to the next level and actually developed a model from this plant height to be able to interpolate individual flights or individual dates of plant height in between flights. And from this, we were able to apply some genomic markers and do QTL mapping. And we saw something really surprising, which is there are a number of QTL that show up early in growth, for instance, that are actually fairly difficult to detect by the end of growth. And so this can partially explain some of the hidden heritability issues. The larger effect QTL at the end masks these ones that are really important early on. And so this will help us break down, or has helped us break down, some of the important genetic effects that occur throughout growth. And similarly, we had a couple near isogenic lines that we knew segregated for a major plant height gene. And for most of these figures, it's all the same. It's just different flight dates over time, and then some phenotypic trait or some measurement on the other axes. And we found that we had non-significant effects at the end of growth when we normally would have measured these with a ruler, but we actually saw that these alleles that are significant for increasing plant height express very early on. So that's something that we never could have detected practically before we had these drone tools to be able to start estimating things like plant height throughout growth. So the largest data set that's been published to my knowledge so far on this, Jacob Washburn collected 43 flights throughout growth. And so you can see, here's just a growth curve that happened over these 1,000 hybrids. And this starts to get more difficult to manage the data for. I mean, processing 40 flights takes at least 3 weeks for my group. And so now as we start to try and get more data, that creates a bunch of different bottlenecks in our pipeline. Okay, so I talked about structural data first. Spectral data is the one that we're finding to be especially exciting. So historically, NDVI, the vegetation index, is used, or one or two other vegetation indices. What FieldImageR allowed us to do is with these data sets, be able to calculate 35, 36 in red, green, blue drone flights, and then up to 90, I think, in multispectral drone flights. And interestingly, some of them are correlated, but each of them do add additional unique information that's useful in our downstream prediction. One of the challenges, though, is how we integrate this data. And I'll show you this later when we start talking about integrating across environments. So these are just a number of vegetation indices over flight dates and 2 different years. And you can see, just using it as an example, vary here, which is a vegetation index. The growth curves or the vegetation index curves are very different between 2020 and 2021. And so how do we actually make sense of this data and use this and integrate it over years? That's been a big challenge, but I think we've made some big gains in the last year with functional principal component analysis and some Gaussian models. So a little example of a trait that's somewhat of interest and that we can really use to demonstrate this data. So plant senescence and southern rust. So southern rust is a disease that comes into maize. It obviously makes some rust-looking stuff on the plants. And so can we detect that via drone? And then senescence is really important, as I'll show you a little bit later, for the grain filling period and when those plants start to dry down and can no longer fill their grain. So one of the things we found by collecting this temporal data over large sets of segregating populations is that senescence was fairly highly negatively correlated to yield. We actually found that grain filling period, or the difference between senescence and flowering time, was actually even more highly correlated to yield across various experiments. So this really got us excited because I can't, as a breeder, go and score all my plots for senescence every day. I can pretty easily fly a drone over all my plots and then process that data to estimate the senescence. So I think this figure has been shown in a couple other slides, but we use variance components to determine how good these measurements are. So variance components are just like heritability or repeatability. They break down replicated trials into saying where the sources of variation are coming from. So when we put all the data in there, obviously most data comes from the flight date. So plants early in growth look different than plants later in growth. So that's where most of the source of variation is. The genotype looks fairly small here, but that's actually pretty meaningful from a breeding perspective. We also have some field spatial variation we can capture, the range row effects, the replicate effects, and then we have some residual. Probably the most important thing to draw your eye to, though, is the temporal repeatability or heritability, essentially the same calculation. So the heritability of most of these are somewhere between 0.25 and then across other studies up to 0.75. But the key is that we have so many of them, right? We have this measurement of temporal, so each day, and then we have all these different vegetation indices plus plant height, and all of those have some heritability. We can combine all those to do prediction. And so for predicting in rust, for instance, we took all of the data that we flew, all the various flight dates, all the vegetation indices, the plant height, and put them in various machine learning models. So you can see the linear model performs fairly poorly, as expected in cross-validation because it's overfitting. But the rest of these, like the k-nearest neighbors, lasso, ridge regression, support vector machines, all of them do a pretty good job in predicting the rust of these plants. So that might not be surprising. You'd say, hey, we can see in the drone that from the drone images, those plants are getting rust. But the thing that's really surprising is that, for instance, the most predictive was actually 17 days after planting using this vegetation index RCC. So that says that these plants, we could tell whether or not they were going to get rust very early in the growing season, right? I mean, there's some other stuff that comes in later, and all these help each other to predict ultimately the rust content of these plants. I'm sorry, this is senescence. The senescence time of these plants. But if we just looked at any one of them and treated them as a phenotype, it would actually be fairly non-predictive. So the phenomic aspect, or using all these various measures that we don't have a priori an idea that it's going to be predictive, is really where the value comes here. So modeling, I mentioned modeling for plant height. That's fairly simple. You know, the plants grow in a fairly predictable Weibull fit model, and then they're done. But something like grain filling period is a little bit more challenging. So grain filling period, again, is the days to silking or the flowering time to the days to senescence. And so there's that period in there, and that's the period that the plant can be filling photosynthase into the grain. So we look at the drone data, and the drone data is a little bit noisy. But Alper here has found, I think this is the mechanistic growth model, found that can smooth the data enough. And then from that, we can actually interpolate a fixed senescence score and compare genotype to genotype. You know, what day did it actually reach 85% senescence? That would not be possible with my regular phenotypic measurements of, oh, this plant has senesced today, but then I can't go back to the field to measure all those plants for another 2 weeks. So that's the type of thing that drone data is allowing, but then being able to model that is really critical for making inferences. So just scoring senescence is a pain, and this is the only deep learning slide I'll show. But Cody and Luke scored 22,000 plots across Texas, Wisconsin, and Missouri from drone imagery, and we're able to develop a deep learning model that could actually give us our senescence scores. So if we go back to our prediction, these senescence scores are the things we need. These are our dependent variables. So we can use deep learning to create the dependent variable phenotype that we need, that then the phenomic measurements can come in and help us explain how it is that what traits or what phenome features create that that phenotype. So it's not just senescence and rust. Alper found a while ago that the most predictive for yield, as we look at all these vegetation indices over all these flight dates, actually was occurring before flowering time. Now, every physiologist would tell you, well, that doesn't make any sense because grain yield is really dictated during that grain filling period. But what I think is going on here is that plant breeders, listening to the physiologists and looking at plants, always look at plants after they flower. Before they flower, you can't really visually observe all that much. And so this is probably the period where there's the most segregating variation, which we can now select on. Previously, we just couldn't see it. Now with some of these drone-based phenomic tools, we can actually see it and predict based on it. So I'm going to switch gears a little bit just to talk about near-infrared spectroscopy. This is another phenotyping technique that has been used for a very long period of time. We use it. We use a thermoanterous Fourier transformed NIR. So we scan all our whole kernel corn, we grind it, we send it for chemical analysis, and then we develop these partial least squares calibrations. Now, the thing that's relevant here is this can scale to that high throughput phenotyping, the increased measure, even if the accuracy is lower. But also we can predict genotypes that we don't have the money to send for chemical analysis. And what's really interesting here is I show you two figures, crude protein and phosphorus. Crude protein has a very nice calibration. The predicted and the actual values of crude protein are predicted very well in NIR. Phosphorus, on the other hand, looks really noisy. But what's interesting is once we apply this to all of our breeding samples, those 17,000 plots, we actually find similar genetic variance between both these measures, right? And we find, you can't see it here, I think this is 42% and 38%. We actually find less unexplained error in the phosphorus calibration, right? And this really was that germ and an idea of we need to focus on high throughput, not on high accuracy phenotyping, because we can make much better decisions from that. So I'm going to shift a little bit to phenomic selection. So the idea behind phenomic selection, there was this brilliant paper by Renaud Vincent that came out in 2018 in G3. And the idea was to take near-infrared spectroscopy data. So this data, which produces these spectrum, absorbance peaks at various wavelengths, and use that as to replace genomic markers. They found that phenomic selection was as accurate as genomic selection, but cheaper, and they used both wheat and poplar to do so. So we happened to have a data set that we'd scanned in for NIR. We had already done a GWAS study on this. And so Holly Lane in my group, along with Jose Crosa at CIMMYT, tried to use and replicate Renaud Vincent's results. And sure enough, we found that we had excellent prediction of yield, right, of grain yield, based on the NIR spectra of the grain that we measured that was harvested from these plots. Now, starch and oil and protein explain some of this. But the overall predictive power of using the phenomic approach, all these various bands on this grain demonstrated that there's something really useful and predictive of this to predict our dependent trait of interest, yield. Furthermore, it worked better across environments and across genotypes than genomic methods did. So Alper has taken this since. I don't remember when this was published, a year or two ago. I'm sorry, the thing's cut off. But he looked at using our drone flights and all those phenomic measures and seeing how that compares to genomic prediction. So there's three main figures here, genomic prediction, temporal phenomic prediction using multispectral data, and temporal phenomic prediction using RGB data. So in genomic prediction, we had about 153,000 markers. In multispectral, we had about 1,068 phenomic features, so a couple orders of magnitude less. And then in RGB, we had even less. So for some of these categories of these cross-validation models, we see that, for instance, CV2, temporal phenomic prediction of multispectral actually beat genomic prediction, right? We see that the RGB prediction, even though those images tend to be better than the multispectral, had less predictive power than the multispectral. And so that gives rise to the idea of we really need to increase the number of phenomic features. If we can get up to 153,000 phenomic features, I think we'll be really doing a good job of predicting whatever trait we might be interested in. So this was a really, really exciting result. I don't know that I've seen anybody else show this yet, but using drones in this way has been sort of slow to be adopted. So let's talk about why this works or how this works. So as a breeder, I'm usually focused on quantitative traits, complex quantitative traits. If we look in the literature, if we look at, where the big press is, it's usually for a single gene, right? And those work really well when they're in a pure additive model with just genetic effects. As soon as we start seeing additive and epistasis or genetic by environmental interaction, we start seeing genomic prediction being needed, right? Or genomic prediction and environmental modeling being needed. I think the niche for this phenomic selection approach is where reality is really messy, where we have a lot of G by E, we have a lot of additive by genetic background epistasis, and other complex things that are modifying the phenotype beyond just genetic determinacy. So how can I explain this in a way that, why does this make sense? This was work that was done many, many years ago by Natalia Cruzado. And what I'm showing you here is a number of days after planting, and then NGRDI, which is just a vegetation index. The dotted lines are three presumably unrelated hybrids because they're from three different companies. The other four lines are hybrids from my breeding program, and they all share a common parent. And I think what you can see over time is that the lines that share a common parent have a similar phenomic trajectory or vegetation index trajectory as opposed to these unrelated commercial hybrids, right? So that makes a lot of sense. Things that are related should look similar. So let's go to the example of humans. So this is my family, my sister and I, my parents, when we were kids, and then later when we were adults. So if you think about your sibling or your relative, you probably share a lot of resemblance. But if you were to ask a stranger, why do we look similar? They'd struggle with those phenotypes we traditionally think of: hair color, eye color, but we know that's not the resemblance we see. Another example is if you think of somebody with Down syndrome, you look at somebody with Down syndrome and you can recognize they have Down syndrome. But if you were asked to explain that, you'd really struggle how, right? Another important component here is that my sister and I look similar at this stage. We look similar here, but we have different phenomic features or facial features over this time period. And those are as we interact with the environment. So we know that siblings that look similar, siblings that are genetically related, raised in the same environment, perform similarly. It's not perfect, but if I was athletic, my sister would likely be athletic. If I was smart, my sister would likely be smart. Similarly, we know if we were separated at birth and one of us was raised in a war zone, we'd probably be 10 IQ points lower. We'd probably be 6 inches shorter, right? That's the G by E interaction. And that's something that just is not captured with pure genetic data. So this phenomic data is actually trying to figure out models that can capture all of that information and predict not just nature, but also nurture. And so that's really, I think, what's going on with this phenomic data we're capturing. We're capturing the genetic component, but also the environment component and the G by E. And we're doing so as those organisms interact with their environment over time. So another way to think about this is we have all these various components that contribute to an end phenotype: DNA, environment, G by E, management, microbes, stochasticity. When we focus on a phenotype, we usually find that something that we care about, but also that is practical to measure, right? Like we don't know even with a ruler, we don't know the exact height of each plant, but we know that using a ruler is pretty well approximates the plant height. So I think over time we've learned that genes interact in networks. They have post-translational modification. There's environmental interaction. And so what we're trying to do, I think, is measure all these easy to measure things throughout all these complex interactions. And then we can put those in machine learning models and then use the difficult phenotype as our dependent variable to predict that. And I think that's really where this phenomic approach is being successful. We focus on what's easy to measure, not just on specifically one thing. So the sort of the challenge I put to everybody then is, we saturated the genome. How close are we to saturating the phenome, right? We went from, in 2013 in maize, 681,000 markers to in, what was this, 2022, 12.2 million SNPs over 1,500 inbreds. So once we get to that type of power in phenomic features, I think we're really going to have some game-changing science. So the rest of the the rest of the talk is just thinking about expanding this beyond different environments. So most of what I presented, we've chosen to focus here in College Station because we have a breeding program. We're collecting a lot of data, but how do we start to think about integrating this to make bigger decisions over bigger, bigger biological and agricultural questions? So the Genomes to Fields project is a great example of this. So we have many different collaborators. We've now done 180,000 field plots, 2,500 corn varieties, and 162 unique environments. Unfortunately, only some flew drones. But once we start thinking about processing this data, this is where we hit a bottleneck with drones. We had an AG2PI project that allowed Mustafa to calculate all of these. So these are the various environments. Not everybody used ground control points or RTK drones. So only some of them could get plant height or structural information, but all of them could get vegetation indices. And for this group, I think the important thing is to start thinking about these file sizes. So this is orthomosaics and plant height cloud files, and then all files, including the raw images and AgiSaw files. One location is, between 0.5 and 2 terabytes of data. That's when we start thinking about combining over every field environment that's being collected, 162 of them, this becomes an insurmountable amount of data. So this is a big challenge. And how do we make this findable, accessible, interoperable, and machine readable? All these are really unanswered questions right now. And we don't even have a repository to store this. We were storing some of this on Cyverse as they changed their business model. There is no NCBI equivalent for this type of data. So it's very expensive to store and share and make public. I mentioned before a challenge of lining up flight dates. So here's 3 Texas environments: Nebraska, Missouri, Minnesota, Delaware, Michigan. People don't fly at the same time. Even if they did fly at the same time, the plant development at that stage may differ from location to location or year to year, environment to environment. So how do we actually integrate it so that day 19 matches up to Missouri's day 17? And so that's where some of that modeling comes in that I mentioned before. We're now scaling this to the Circa project. For those of you who aren't familiar with this, circular economy that reimagines corn agriculture, it's about really better utilization of nitrogen. So this is not going to be as simple as doing some GWAS or doing some phenomic selection. This is a lot more complicated about shifting the entire growing period of maize, utilizing biological nitrogen inhibition. There's a lot of really difficult concepts as we start thinking about how we can make corn better utilize nitrogen, be more, have less greenhouse gas emission, less pollution, and less farmer applied nitrogen. And the reason I wanted to talk about this is because this is really the first transdisciplinary project I've been a part of, where we're going from genomicists who do evolutionary mining of alleles to people who are doing gene editing, to the physiology, and then integrating this crop modeling component, both at the landscape scale and in the individual plant scale. And now as we start talking about this data, figuring out how to integrate it becomes really, really challenging. And so I guess the last project I'll mention, and Sushma was a great part of this, was a project that DARPA supported to look at how would we detect a new pathogen. So we know existing pathogens. You might be able to collect field data. You might be able to find genes that for resistance. But if we had either like a COVID type new pathogen that comes in, or if some nefarious actor tried to introduce a pathogen to disrupt our food system, how would we detect that? And so the idea here is that we use the plant reactome to identify common shared pathways that we can detect disease from. And those diseases could be viruses, bacteria, fungi, whatever. We then would need to use proximal sensing to connect individual networks, metabolites, to something we can measure. But ultimately, there's this big gap, not only in the reactome and connecting the reactome to proximal sensing, but then extracting that to the landscape scale. If we're really looking at a disease monitoring network for diseases we don't know exist yet, that may share a common trigger pathway in these plants, how do we scale it up to drones, which can do farm or maybe county scale, to satellites, which can actually do global scale? And it turns out this was a really tricky problem. and the data just doesn't really exist to solve this. So we tried using our robotic greenhouse with a multispectral sensor to try and do some individual plant collection, some Raman spectroscopy. I'm sure Sushma's talked to you about the plant reactome a lot, and so trying to link that in. And finding individual compounds using Raman spectroscopy even is a challenge because of the water bands and because how those compounds that are in disease pathways may interact in planta. So we couldn't go through an entire field or the entire US crop, extract DNA and figure out if it's got some disease, right? So connecting these for a food security standpoint is a really tricky and difficult problem. And photosync is another type of phenotyping that we used in this project. So I think, as we think about phenomics and phenotyping, it's not just drones, it's not just robotic cameras, it's not just NIR, it's some of these other sensors that so far we don't really know how to connect this all together. And as we think about all these types of data, the goal from my perspective is let's increase the dimensionality. So we can increase it spectrally, we can increase it temporally, and we can increase it spatially. And as we think about spatially, Aaron, I'm not going to go through all this, but he now has started phenotyping from drones individual plants. So everything we've done is on a plot basis. We have 70 to 90 plants in a plot. As we start thinking about phenotyping individual plants, you can multiply those data sizes I just showed you probably by an order of magnitude to get at individual measures for individual plants. And my last data slide is just thinking about what the future looks like in plant breeding. So we're going to have dependent traits of interest, maybe their yield, but maybe their nutrition or ecosystem services. These are our phenotypes. We're going to have some physiological measures that people have routinely taken. We're going to have some temporal phenomic measures. We're going to have some segregating remote sensing phenomic features, and we'll probably still include some genomic measures. And somehow we have to integrate all this through growth models and weather data to be able to predict the best untested hybrids for untested environments. And this is a really, really tricky challenge, but I'm super excited about the future and all the various disciplines that are working on this problem. Just knowing this group, I threw in, we're not the only group doing UAS phenotyping. There's a lot of different groups across the country and across the globe that are all focused on this. So I'll end there. Hopefully I'm not too far over time, but happy to take any questions or discuss any of this further.
Thank you, Seth, for this wonderful talk. And we can take the questions now. If anybody like to ask question, please raise your hand and you can unmute your mic. Peter?
Hey, Seth. Yeah, thanks. That was awesome. Do we so just talking about data sizes, and connecting to the genomic, making the parallels with genomic data. As the technology advances, we figure out which pieces of data are crucial to keep long term and which pieces can be, discarded over time. Do you have any sense for that yet? Do you have you want to speak to that at all?
Yeah, going back to, I'll go back to the slide and just thinking about some of these. So we actually had part of our AG2PI project, we were supposed to have a workshop on this. We never quite got that far, but we still hopefully will in the future here, about what is, the minimum amount of data to retain. So the minimum amount of data to retain would probably be the raw images, because you could recreate most of these things if you knew the specific process. But the raw images plus the shape files and the field book telling you what's in each of those images, that would be the bare minimum. But if we think about reproducibility, it's really going to be probably those tabular data features, like like this, where we have various plot IDs and the various vegetation indices. That's probably going to be the most reproducible because, getting to that from raw images, there's a lot of variability. And all those methods are going to continue to improve. So I think it depends partially what the goal is. Is it reproducibility? Or is it as, let's say we develop a new algorithm to predict northern corn leaf blight. Well, if we have this data, it'll be really easy to plug that in and predict northern corn leaf blight. If we have raw images, we're going to have to go through that entire pipeline again. So I think it's really important to discuss, what is the goal? Is it reproducibility or reusability and discovering new things? Does that make sense?
Yeah,
Thank you,
definitely.
Doreen.
Thank you.
Yeah.
So actually, I was going to follow up with Peter. I mean, I'm thinking about images and sequencing, and how do you come up with what you're showing here, making a decision at what point in time the group feels comfortable with basically extracting out the equivalent of the AGCTs to this format, and how much of the old data do you really need to go back to extract this out in the future versus the new imaging and the sensing technology that's coming along? Because I think it is going to be really critical when you were talking about the storage space, because I think that I have a group of people in my office with me, because I think that moving forward, when you're talking about those AG2P projects, if you think about, if we have these for 15 commodity groups, right? Really is like the sequencer. How do we make a decision? How does the community come down to a decision to get to this extracted form? And when do you feel like you're losing too much if you get rid of the images?
Well, I think we need another 10 years before we make that decision. So if you think about genomic data, I can always take that individual and go resequence it, right? But this phenomic data was specific to that environment. And so there's no way ever to go back and collect that. So if we get rid of it, that's a problem. The other thing is we look at these vegetation indices. So, the thing that I don't think I've still wrapped my head around is these are relationships between three bands for RGB, right? There's some multispectral in here, but it's various relationships. Talking to the statisticians, there is an infinite number of relationships. So we may still not have found the best vegetation indices or the most unique vegetation indices. So if we got rid of the raw images, if somebody developed a new method in a year that's like, this vegetation indice is way more predictive than anything we had in the past, we couldn't go back and extract that. Right? And unlike genomic data, we can't just recreate that. We can start collecting new sets of data going forward with that vegetation index, but we can't go back to all the data we've invested all this money in, over the last 8 years. It would be gone. Does that make sense?
No, I agree. I think it's really a matter of coming up with what's going to be the consortium model for us in agriculture to share the information.
Yeah.
Thank you. Leonore?
Oh, I'm sorry, I forgot my hand was up, harping on that same sort of issue. I had to go and look and think about, like, what do other projects that generate mondo amounts of data do? So my neighbor, for example, works at Breakthrough Listen, and they collect like a petabyte a day and they keep it. He spends his life like building server racks, and they're keeping all of that information. But if you look at CERN, they filter things through. They're deciding like they have some sort of consensus agreement about that. I'm wondering about, whether or not there are technologies that enable us to compress the data, and, would it be at a good enough fidelity where we would feel comfortable to take that and, sort of crunch it down a little bit more? I don't know. I mean, I'm sort of thinking we need to maybe look at physics and, these other like Mondo data projects and sort of see what they're doing. But so my other question was also around like the metadata that we need to keep. Like, do we have a standard on that? Not yet.
Well, I mean, so the first data set, I'm obviously biased, right? But the first data set that was made fair and publicly released was our genomes to fields data set from 2017, which we put up on Cyverse in 2018, I want to say. And we worked a lot with the Cyverse folks and thought a lot about what metadata needed to be in there. If we're going to redo that today, I mean, Genomes to Fields is actively trying to figure out what we're going to do on this. There's different file organization, different metadata that we would, have now. And I think there's still just so many open questions, and quite honestly, there's so few people collecting this type of data. There's a lot of people who have been flying drones over their field and they put it on a hard drive and they haven't analyzed it, right? There's many fewer of us who've actually been working on developing these pipelines and crunching from, especially from investigators across the country. I mean, wheat, cotton, and maize, I think, maybe soybean, have groups where they've done this. Until you start, each person has a different drone, each person flies different days, each person believes something's important that somebody else doesn't. Until we get a more of a critical mass, I'd be really hesitant to say, well, this isn't important, if that makes sense. Again, I feel like we're at 1987 or 1988 with QTL mapping with where we are in this. So, there's a long road to hoe.
Thank you. Rex?
Hi, great talk. I was wondering what you think the impact of the constant improvements in sensor technology, and how would that impact reproducibility and the value of those, quote, old data sets?
That's a fabulous question. We just got a new drone. So we love DJI Phantom 4. We can have an undergrad fly it, $6,000 to $12,000, depending on what features. We had to buy a US made drone this time, $58,000. And even my grad students can't figure out how to fly it. But it has a different sensor. So we're not really sure if we had been successful, how we would integrate that. When we think about the Genomes to Fields project, different people had different sensors, and the red band is not a single spectral band. It's a range of spectral bands, and that differs from camera to camera, and especially when we talk about NIR or far red. So that's going to, that spectral range is really going to play a role. Resolution, I don't think will play a role. I think historically, we've been downsampling resolution just because it's so much data. But the spectral features, the actual bands it collects, may make it difficult to integrate this in the future. But I don't think we still know what the best range of bands are, right? There's a balance between the cost of the sensor versus the data it collects. So I don't have a great answer for you, but it's complicated. How about that?
Thank you, Seth. John?
Trevor's been waiting longer than me. Go ahead, Trevor.
Thanks. Hi, Seth. Great talk as always. So you highlighted early on that there were differences in what was detected when you're using different drones, but different drones is just that first step. And then there are all of those other steps in the analytical pipeline. Have you looked at variations or different tools for each of those steps to see how swapping out different technologies affects your end result, different, orthomosaic approaches, things like that?
So there have been a few people who have done stuff like that. We did a little bit early on. Let's go back here. So I think the biggest one is what you use to orthomosaic. So Metashape versus Pix4D would be a great example. And then there's some open source tools, but they don't work nearly as well. The challenge we have is my group is ultimately interested in end decisions. And so if we chose this pipeline, I don't know, 2 years ago, and to be able to compare all these different locations, we just sort of have to stick to it. So there's probably a better method for each of these. There's new software coming out. There's other groups working on it. But to be able to compare the end product, you would really have to use the same methods throughout all of it. And for those, 20 or so environments that I showed you, that took over a year of Mustafa's, Aaron, and Alper's time. So we're really hesitant to swap out one for another and see, well, maybe it works better. Well, if it does, we're still not going to go and reprocess all that data at this point. So I think these tools in the pipeline will get much better. I mean, we think back to QTL mapping, and the early algorithms really didn't work all that great. And then over time, as more people developed better algorithms, or genomic selection, same thing. There were steady improvements. So my focus is on analyzing all the data jointly. But there are other groups who are, asking, does this drone work better than this drone? Does this metashape work better than Pix4D? Does that answer your question, kind of?
Yeah, definitely. Thank you so much.
Okay.
John, you can ask.
Yeah. So following on that, I put this comment in the chat. Seth, good to see you again.
Likewise.
I haven't been following all the funding line by line, but is there anything real or possible of getting more funding for these things into USDA, like we've talked about for years? Some kind of NCBI-like program?
Yeah. Great question. Yeah, I don't remember which papers we were on together, but I know there's been at least 4 or 5 papers that I've been a part of that says we really, really need these resources to store these things. I think currently we still get a lot of pushback of, are we just collecting data to collect data? Right. And that's why I focus so much on the case studies, not in this talk, because this is more of a data group, but focus on case studies showing how these tools have opened new research areas and really been helpful. Because if you just say, I want to collect, saturate the phenome and collect 1 million features, that sounds like a fundamental research project, right? And doesn't, it's harder to make the sales pitch. But as we show that these things can really be transformative to our science, I think maybe that demand will increase. So
yeah,
NSF is hosting something, I guess next week, that maybe some of this group is a part of, looking at next generation animal and plant breeding. I think that'll play a role. I ultimately think it's not going to come from NIFA, right? It's going to either need to be ARS, maybe part of the Egg Data Commons, maybe.
sure, sure,
or just separate funding for this type of database.
yeah.
But so far, with all of our findings, we can only say that this works really well in College Station. We're just getting to the
Right.
point where we can integrate data across other locations. Maybe it doesn't work in Iowa. And if it doesn't work, I'd hate to say everybody should be collecting this data.
Sure. No, and I appreciate that. Yeah, I think I think you're spot on. And some of the things we're talking about on the board on ag and NCFAR and FFAR is, how do we get some better interagency cooperation? Because NSF is doing some of this, getting started, whereas USDA, whichever group it is, isn't. And I agree. I think that message that you said, what's the point here? What's the end user? It's not, you could make arguments about NCBI, but they do have medical purposes or those types of things. So thanks for that. Hopefully we can keep that rolling forward.
Doreen,
and I
you
don't
had
think
something? Yes, sorry. Sorry, Seth,
let me just add real briefly, again, going back to 1986, if you talked about what genomics could be used for, that looks nothing like what it's being used for now, right? I mean, people are going to keep, but saving this data once it's collected so that, there's a library to go back to, I think is really important.
Doreen, you had something?
Actually, it was already kind of beaten to death, but from my perspective, Seth, you guys, I see NIFA as the extramural arm that's driving research projects, that's demonstrating the utility. And then, as you mentioned, the National Ag Library. I think that there should be a discussion between NIFA pushing to USDA about the needs and the demonstrated ability. And I don't know, I know that everybody's tired of white papers, but given everything that's come out of the AG2PI, like the genomes to field, I think it's time for that group, if they haven't done sort of a summary, sort of best practices, interest in requirements, right, that have come out of it, this would be the time to do it, because, you know, that's how the agencies drive either an RFA or an ask, right? And I think you guys, as the individuals who've done this with the genomes to field, you know, maybe tied with some of the things that Ed's doing right now, and these examples would be a really nice time to have like that white paper go to maybe CSSA, as, you know, as the Crop Society of America, and as we're doing the bioeconomy initiatives, which, you know, NSF has the tips from, saying we do need this space to operate. We do need a location for this. We know what the challenges are with cybers and why there have been issues with now you have to change that. We're experiencing that as well. And so perhaps there could be some sort of discussion with the NSF as an agency, as where Cyverse was initiated more for supporting some infrastructure things, that maybe there can be a link with the bioeconomies and the requirements there and what's come out of this to target that. I'm just kind of throwing that because I think that without that white paper specifically stating it, it becomes harder for the agencies to make the ask.
Thank you. Thank you, Doreen. Thank you, Seth, and everybody else. So we are just 1 hour of the webinar, and I wish you get back well soon, Seth. Thank you very much.
Thank you all for having me.
Yeah.
Thanks for inviting me.
Thank you. Thank you, everyone.